Begin a look today at our only liquid connective tissue, we have the blood. So it is a connective tissue. It does have cells, it does have a ground substance to it, and it does have proteins which become the fibers that are found within the blood as well. These ones we're not able to see the majority of the time as they are dissolved in the plasma itself, and the plasma becomes the ground substance portion, so the non-living component of our connective tissue. The cells that are there are formed by hemopoiesis or hematopoiesis. This is the actual process itself, and so this actually begins not in the blood itself, but in the red bone marrow. And so we see this process beginning within the bone marrow itself, rather than being in the blood where they are being formed. These are being formed by stem cells called hemoblasts. And so these cells are going to produce not just the red blood cells, but all of the white blood cells and our thrombocytes as well. In terms of what we use blood for, primarily we think of blood as kind of being the transport process. And so this is where we're going to get all of our nutrients, all of our waste products to and from the tissues themselves. And so any nutrients that we bring into the body needs to be able to go through the bloodstream to be able to get to the tissues. Any waste products that are created goes through the bloodstream as well in order to get to the kidneys to be able to be gotten rid of as waste in the urine. But it also does some other things for us as well. It helps to do things like regulate body temperature. And so by either bringing blood to the surface of the body towards our skin or keeping it away, we can maintain our body temperature. It has an ability to buffer, and so we can change pH levels within the body. And as we either drink or don't drink, fluid levels are going to be altered, and so we have the ability to help with that process as well. And then anywhere where we have a problem with the immune system, we're going to utilize the blood in order to get that there. So body temperature, this is essentially because water does have a great ability to transfer heat, and so with that water in the plasma, we can transfer heat either out to the surface and get rid of the body heat, or we can keep it cooler down inside as temperature cools off. So in summertime, we're going to, or with exercise, things like that, as body temperature rises, we're going to dilate all those blood vessels that are near the surface of the body. This is why skin starts to blush as we get warmer. And then as we cool, we're going to pull that blood away from the surface, get it down deeper into the body, and conserve the heat. pH levels comes from the ability of the blood to buffer the blood itself. And so pH normally is in the realm of about 7.4, and it's either going to utilize the movement of carbon dioxide to or from the body in order to maintain that pH level that we want. And so we're going to drive it towards the lungs, we're going to drive it towards the kidneys in order to help to maintain a normal pH within the blood itself. As we consume fluids, so if we're drinking a lot, we're going to have excess amounts of fluids, and so if we're constantly drinking throughout the day, we're not necessarily needing all of that fluid that we're bringing into the body, and so we can get rid of it from the body that way. Same thing if we're not drinking a whole lot throughout the day, we can conserve that water and keep it into the bloodstream to maintain blood pressure. That's there. And so within that as well, we also have the exchange of fluids between the actual bloodstream, so inside our plasma, and the cells around. And so it goes through the interstitial fluid itself. And if that fluid goes out there too much, we get things like swelling. If we need to pull it back in, we have the ability to do so as well. And so depending upon where we put that fluid at any given time, if it's too much fluid out in the tissues, we're going to get swelling, we're going to get edema, things like that. If there's too much fluid inside the blood, we're going to get high blood pressure. Blood pressure is going to start to go up and up and up, and that's not a good thing either. So in order to maintain this normal fluid balance throughout both the extracellular space, the interstitial fluid, and that of the plasma, we utilize proteins. And so primarily proteins are going to be the big thing that's going to maintain that fluid balance between the interstitial fluid and the blood plasma. And then we also have things like salts. And so the salts, if we're losing salts through things like sweat, we're going to be losing them through the blood as well. If we should happen to encounter a foreign antigen, we have the ability to initiate an immune response. And so leukocytes, or our white blood cells, are going to be able to mount an attack if they need to. And so in response to signals being sent by other immune cells, part of the immune process itself, they're going to be drawn to those areas where we potentially have infection. From there, we're going to be able to deliver things like antibodies. And so if we've come across that infection in the past, we can deliver those antibodies into the area where we can then eliminate the infection potentially before it causes us any problems once again. If we should happen to have damage to a blood vessel, this is where blood clotting comes in, and this is where our thrombocytes, our platelets, are going to become active. And so they're going to essentially become attached onto the connective tissue of the blood vessel, and at the same time they're going to send signals saying that there is a need for more and more platelets. So when we look at blood and we look and see what's inside there, we'll notice that we have kind of some different components that can be found within the blood. And so if we take a blood sample and put it into a test tube, and then put it into a centrifuge and spin it down, what we're going to find is that it separates into three basic layers. And so we have the layers of plasma up at the top, and so this is basically all water, and so this is what's going to be sitting at the top there. We have a layer of redness at the bottom. This is all of our red blood cells. And then we have a thin little layer called the buffy coat, which sits in the middle. And so this is just a small little portion there. So the majority of blood is actually either the plasma, the liquid portion, or red blood cells. And so erythrocytes make up somewhere in the neighborhood of 44-45% or so of the blood. 55% or so is the plasma, and then only about 1% or so is the actual buffy coat, where we have all of our white blood cells and all of our platelets. And so not a whole lot there as far as white blood cells and platelets goes. It's mostly all the erythrocytes and the plasma that are there. But we do have a difference as well between what we call formed elements and the non-formed elements. And so the plasma contains all of the non-formed elements. And so the plasma has everything that is not a living cell. And only the buffy coat and red blood cells are our actual formed elements that are there. So the erythrocytes, the white blood cells, and the platelets become all of those formed elements that are there. When we give blood as a donation, we can separate the blood out into these elements as well. And so we can break them down into the different components that are there and utilize each one in a different way. So the formed elements themselves are being made out in the bone marrow, so they're not initially starting in the blood itself there. Erythrocytes themselves are the majority of all of the formed elements. So they already made up some 45% or so of the actual blood sample that we had there, and only 1% was the white blood cells and the platelets. So in terms of all of the formed elements, the red blood cells are going to be somewhere in the neighborhood of 99% of all of the formed elements. They're going to be the majority of everything that's there. From there, we then have leukocytes, our white blood cells, and our platelets. And so platelets make up a little bit larger quantity to them, and leukocytes are then the lesser of the three different types that we have there. And so defense against our pathogens are our leukocytes, blood clotting are our platelets, and every day we're damaging small little blood vessels here and there, and we need to be able to clot those out. When we look at blood, that 45% or so component that's there, is looking at what we call the hematocrit. And so sometimes this is referred to as the crit. The hematocrit is the percentage of those formed cells that we have in the blood itself there. And so typically in terms of males, kind of the number that most labs are looking at somewhere in terms of being perfectly normal is in that 45-degree range there. For females, it's usually typically somewhere around 40 as being that normal range that they're looking for hematocrit itself. As the environment changes and as things around a person kind of change, the hematocrit can start to go up or down depending upon that. So this is primarily dependent upon our ability to deliver oxygen to the tissues themselves. And so if we don't have the ability to get that oxygen to the tissues, we're going to compensate by making more red blood cells. We're going to make more and more red blood cells until we can get that oxygen level there. One of the kind of quickest and easiest ways to see this effect is simply by changing altitude, by going from, say, a relatively low sea level altitude all the way up to the mountains somewhere. And so going from like the beaches in California all the way up to the mountains in Denver, all in one flight. And within a short period of time, your body is going to be looking for more and more oxygen, and it will typically start to build more red blood cells. And those red blood cells themselves are, by all definitions, don't really meet the definition of a cell. They have no nucleus, and so there's no nuclei inside the cells themselves. The majority of the organelles are gone. It is basically a big disc filled with a very large amount of hemoglobin. And so the job of red blood cells is delivery of oxygen to the tissues. And so it's going to deliver O2. It's going to pick up carbon dioxide from the tissues and bring it back to the lungs. But the cells themselves are geared towards that process, movement of oxygen to the tissues and picking up CO2 and bringing them back to the lungs. In order to do that, they're going to optimize the amount of cells that, or the amount of surface area that can be done doing that by having this biconcave disc. And so the cell itself dips in the center. The cell dips in the center on both sides, and this optimizes the amount of surface area there for gas exchange within the cell. By not having a nucleus there, we increase the ability of that cell again to have more and more hemoglobin within it. On average, these cells only live about 120 days, and so they're being constantly replaced, and so some 3 million or so are being released every single second. So huge amount of cells being formed every day, huge amount of cells also dying off every day. That hemoglobin that's inside the erythrocytes themselves, we've got somewhere in the neighborhood of 250,000 hemoglobin molecules in every single cell. And so we have just a huge amount of hemoglobin available in every single cell to be able to transport that oxygen to the cells. Each hemoglobin has the ability to bind 4 oxygen molecules. So there are 4 proteins in each hemoglobin. Each one of those proteins, we have 2 alphas and 2 betas for the proteins, and each one of those proteins has an iron in its center. So that iron can bind a oxygen. So 250 million, to make the math easy, 250 million hemoglobin times 4 oxygens gets us roughly 1 billion oxygen molecules being transported in each cell every time that it goes past the lungs and then out to the tissues themselves. When we look at blood, if you've ever had blood drawn, typically you're going to see that blood as being a kind of a deep red color. So this was the venous blood that they were taking. But if you ever saw arterial blood, or blood that comes out onto the surface, so if you get a cut somewhere, as soon as that blood hits the surface and there's oxygen available, it turns into a bright red. And so it's going to change color based upon the amount of oxygen that's bound to the hemoglobin at any given time. So here we have our hemoglobin. Here we can see in the very center of the hemoglobin we have our irons. And so our 2 beta chains there, our two alpha chains, and we have those irons at the very center of each one. And with that, we have the ability to bind an iron. And as each iron binds, the next iron binds easier. It becomes harder and harder for the iron or the hemoglobin to resist iron in a sense. And so more and more of that iron binds, and it binds faster and quicker to that. But it is kind of a weak attachment, and so it's going to be relatively easy for the hemoglobin to give up that oxygen once it gets to the tissues. Since those red blood cells only live about 120 days, what is inside them needs to be recycled as much as possible. And so when a red blood cell dies, it's typically going to often get caught inside the spleen. And so the spleen is going to find those red blood cells, it's going to trap them, it's going to destroy them, and it's going to break them down into their components. The liver is also going to help in that process itself. And when it does so, it's going to break it down into iron. So we're going to have iron molecules that are there. If it's in the liver, that can then be instantly stored away inside the liver itself. And so we can store away that liver for another time when we need it. It can get transported to the bone marrow, and we can make some more red blood cells. That's there. And then we have a heme component. And so that heme is going to be available there as well. And that heme portion is waste. And so the heme component is going to eventually get converted into bilirubin. And that bilirubin is going to get sent over to the liver, where it's going to then be lost in the bile of the liver as bile salts. And so it becomes part of the bile component that's there. The last component, the globin portion, is also recycled as well. And so the globin component of our hemoglobin is recycled. All of those amino acids can be used again, either to make more red blood cells or to make something else somewhere within the body itself. And so all of those different proteins that were broken down into the amino acids, those can potentially go out and make new erythrocytes or anything else that we need within the body itself. Red blood cells do have antigens of their own, just like our whole body has antigens on the surface of every single cell. Those antibodies are there to mark not just that they are part of ourselves, but also they become specific towards the type of blood that we have. And so we have what we call the ABO blood groups. Those ABO blood groups essentially means that we have either antigens for A, antigens for B, or no antigens. And so A antigens can be on the surface of cells. B antigens can be on the surface of the cells. We could have both A and B antigens, or we can have no antigens on the surface of the cells. If we do have antigens on the surface of the cell, so if we have an A antigen that's there, and so we have our cell here, and then we have a little A antigen that's there, we don't want our body to make any anti-A. So we're not going to make any anti-A antibodies. But if that A-type blood becomes in contact with, say, B-type blood, that particular cell, as the B-type blood comes in contact with the immune system of that person that has A-type blood, their body may then start to form B antibodies. And those B antibodies can then come and attack that B-type blood. And so we have the potential that they're going to destroy that particular type of blood that's there. So when we look at it, we have A-type blood here. So what we were just talking about is we had A-type blood, so it has antigen A. They have the potential to have anti-B antibodies, but they can't have anti-A because they would destroy all their own blood. B-type blood has the B antigens on the surface of the cells. Those B-type of antigens then can make for us to have anti-A antibodies. So they're going to destroy that type of blood if it comes in contact with it. A B type of blood has both A and B antigens. So they have both antigens, which means that they can't make any antibodies. So they're not going to make anti-A or anti-B because it would both destroy their own red blood cells. And then lastly, we have O-type blood. O-type blood is free of any antigens, but their blood will make both antibodies. So an A type of blood has A antigens, but may have B antibodies. B type of blood has B antigens and may make A antibodies. A B type of blood, no antibodies, so we don't want any antibodies there, and their blood is going to be able to accept essentially all kinds of blood. So since they don't make any antibodies, there's no antibodies here, if we give this person A-type blood, no big deal. If we give them B-type blood, no big deal, because they don't have any antibodies towards them. If we give them AB blood, no big deal. And then lastly, if we give them O-type blood, again, there's no antigens on the surface. They can't make any antibodies anyway. So the AB type of blood is said to be the universal receiver. They can receive any kind of blood. O-type blood, because it has no antigens on the surface, is said to be the universal donor. And so they can give their blood to anybody because there's no antigens on that surface. There's nothing for any antibodies. So even if we give it to A or B, there's no antibodies there for it to react to. So they're not going to react to that type of blood there. The other side of blood, and that you may have heard about in terms of blood itself, is the concept of it being either positive or negative. And where that comes into play is the RH group. And so RH is another type of antigen. And if a person has that antigen, they are then said to be positive. They cannot make any antibodies to it, so we can't make an antibody there. Or they are said to be negative, and they do not have any of the antigens. So there's no antigens on the surface there. So in a sense, the true universal donor would be somebody who is O negative, and the true universal receiver would be somebody who is AB positive. So they have the ability to receive any kind of blood because they won't have any kind of antibodies whatsoever. That's there. If we give the wrong kind of blood to somebody and they have antibodies to that particular type of blood, they have the potential to essentially destroy all of their blood. It goes through a process called hemolysis. So that's going to destroy all of that blood there. And it's the same concept that we use actually to identify blood types as well, so that we have the ability to determine which blood type a person has, is if we mix their blood with a particular antibody, that particular antibody will destroy that, and we'll be looking for clumping to be able to determine whether we actually have that particular type of blood there. The white blood cells, or leukocytes, are designed for protection. And so these are going to defend the body against any kind of antigen or pathogen that happens to come into the body. Unlike our red blood cells, these do contain a nucleus. They contain many other kinds of organelles as well, because they're going to have to make things along the way. These cells tend to be a lot larger than the red blood cells, and so they tend to have a much larger size when we look at them under the microscope. And whereas a red blood cell, in a sense, is kind of a red blood cell, the leukocytes come in one of two types, either granulocytes or agranulocytes. And so depending upon what we can see when we stain them under the microscope. So when we do a blood smear, so if you're donating blood or you're giving blood to the lab for workup, whatever, and they do a blood typing, they're going to take that little piece of blood there, the little drop there, put it on the slide, smear it, and then stain it. And what we end up with is something that we see here in the picture. And so we see all kinds of red blood cells, and then scattered throughout we have some white blood cells, and we're able to then determine each one under the microscope that is there. So our granulocytes are going to be granulocytes because when we look at their cytoplasm, that cytoplasm is going to tend to have granules inside. So we have granules in neutrophils, we have granules in our eosinophils, and we have granules in our basophils. And so they make kind of like dark dots inside the cytoplasm that's there. Neutrophils are some of our most plentiful white blood cells, and so they tend to be at the higher end of the scale there. So we have the majority being all neutrophils. Some 60+% of them are neutrophils. Eosinophils and basophils, we have relatively small amounts of them. We don't necessarily need them as often as far as the cells themselves go. And so eosinophils contain granules inside of them, and so those granules that are inside them are able to help with the phagocytic process that they do. Eosinophils we tend to see with parasitic worms, and so we're going to see them elevated in times of parasite infection. Basophils will oftentimes see when they release their histamine due to an inflammatory response, and so we see them a lot with things like allergies that are there. Our agranulocytes, we have our T lymphocytes and our B lymphocytes are both types of lymphocytes. And so we have two kinds of lymphocytes that are present within them. These lymphocytes themselves tend to be just about the same size as a red blood cell, just a little bit larger than a red blood cell. When we look at the lymphocytes themselves, they tend to have a very huge nucleus. And so that nucleus basically takes up almost all of the inside of the cell itself. These make up the second largest amount of white blood cells, and so they're somewhere in the neighborhood of about 30% or so on there. T lymphocytes actively go out and attack foreign cells or virus-infected cells. So they're actively attacking the infection. Whereas B lymphocytes make antibodies. They're making the substance that's going to be able to bind up those particular infections that are there and stop them from being able to go any further. Natural killer cells are kind of non-specific cells. They go out and they attack anything that is abnormal. And so it may be our own abnormal cell, could be like a tumor cell, or they are infected cells. But they don't really care which type of infection it has to be. The last of our agranulocytes is the monocyte. The monocyte is the largest of all of our white blood cells that we have. They again tend to be relatively low in their numbers that are out there. They are phagocytes, and so they're going to phagocytize just about anything that comes along, whether it be bacterial infection, viral infected cells that have been killed, damaged tissues, so you sprain your ankle, something like that, monocytes are eventually going to come into the area itself. They have a very distinct nucleus that is typically usually U-shaped or kidney bean shaped in terms of its appearance that's there. That's kind of one of the hallmarks in terms of how we see a monocyte under the microscope. Thrombocytes are platelets. These are not actual cells, but they are actually cellular fragments. And so our megakaryocyte makes the cells themselves, or makes the platelets, and the megakaryocyte then, in a sense, kind of, the cell has little chunks that comes off of it. And so little pieces of the cell start to be exocytosed out of it, and those are what go out and become all of the platelets that we have that are there. Platelets are, along with the white blood cells, are relatively short-lived, and especially for the platelets because they don't have the ability to repair or do anything because they're just kind of pieces of cells. The plasma that's inside the blood itself is primarily water, And so water makes up the majority of all of the plasma itself. If we were to spin down the plasma more and take out all of the substances that are inside, we would find that the water makes up the majority of everything that we find there. Essentially, it is extracellular fluid since it is outside of the cells and is part of our tissue that we have there for them. And we can separate out the fluid portions that are there, and we can get the serum when the proteins are taken out. So if we take out all the different proteins that are inside the plasma itself, we can break it down further into serum as well. The plasma proteins serve an important job in doing different things within the blood itself. We have many different kinds. One of the more important ones in terms of maintaining fluid volumes are things like albumins. We have our globulins for our immunity. We have fibrinogens for blood clotting. And then we have regulatory proteins of all different kinds that are there as well. So our albumins themselves are the majority of all of the plasma proteins. They're going to help to regulate water. And so they're going to maintain that water balance between the interstitial fluid outside of all of the rest of our tissues and the blood itself. And so if more of that albumin starts to leak into those tissues, that's going to pull more water into it, and we're going to get things like swelling. The globulins are our immune cells, or our antibodies rather. They come from the immune cells. And so the antibodies that are there, one of several different kinds depending upon what we have as far as the type that is going on within them. The globulins themselves have both alpha and betas. Once again, this is going to bind up to other water insoluble components that can be found within them. We can use that for transport of different substances that may be hydrophobic or water insoluble that are there. Our gamma globulins, our antibodies, we have for defense, and so this comes in one of several varieties as well. Fibrinogen, this is one of our proteins that we're going to utilize for clotting. And so fibrinogen is not something that we're going to use directly within the clotting process. Fibrinogen is soluble in water, so it's floating around. We can't see it. But once fibrinogen gets converted into fibrin along the clotting cascade, this then is able to essentially work like a net to help to build the wall within our platelet plug. And so this helps to stop the blood from being lost out of the blood vessels. Our regulatory proteins, these are everything from things like hormones, and so being sent from one part of the body to another. These can be enzymes within the body itself. Some of them are proenzymes, things that we're going to utilize for even things like the clotting process within the body itself. The different components that are inside the fluid that's inside the plasma that's there, this helps to maintain normal concentration gradients. And so we're going to have higher concentration of certain substances in the blood compared to those out in the tissues. And this is what's going to help to get our substances from the blood into the tissues themselves. And this is especially so for things like oxygen and carbon dioxide. But all of our nutrients run along the same process as well. And so inside the plasma, we have high levels of oxygen compared to that of the interstitial fluid. And so lots of oxygen there. So the oxygen is going to want to go from that higher concentration that's inside the plasma towards the tissues. And so it's going to move from the blood into the tissues. When we're at the tissues, the CO2 is going to be higher inside the interstitial fluid, inside the tissue. And so carbon dioxide is going to want to move from the interstitial fluid into the blood. And so it's going to move from that direction there. And the same thing's going to go for other nutrients. We're going to have a high concentration in the plasma and a low concentration in the interstitial fluid. Those nutrients then move into the tissues. waste products. We're going to have a higher amount of waste products in the tissues, and they're going to want to move into the blood so that we can take it away from there. In physiology, you'll go through this quite a bit more extensively. We're just kind of getting an intro into it for anatomy here.